Hello, hope that everyone is safe. For today's lecture, we are going to be talking about meiosis and DNA replication. And I wanted to start by showing this image of a cardinal. So hopefully many of you are familiar with the mascot of Wesleyan. But normally we see cardinals as being red, at least the males, and then the females are a little bit of a reddish brown, but definitely not as spectacular looking. So we're going to come back to this slide after I talk about my meiosis, and we will discuss why this cardinal that's on the slide is half brilliant red and half of that more muted brown color. Also, before we begin, I just wanted to let everyone know that I will be putting some questions on the slides to help you quiz yourself and make sure that you are on top of all the information, or at least some of it. And I've also linked a review tool here, which will ask you to identify different stages of mitosis and meiosis. This is definitely not required, but I know that some people really do like using it. Okay, so yesterday we talked about mitosis and how the cell normally divides, and mitosis is what occurs normally in a multicellular organism as that organism grows, as that organism replaces tissue that was old or damaged. Meiosis, on the other hand, occurs only in gamete cells, and so for mammals at least, and animals in general, not just mammals. Meiosis only occurs in the germline, or in the cells that will give rise to sexually reproducing cells. So meiosis, unlike mitosis, has 2 rounds of division. It only has 1 round of DNA replication, however. So this is really important to remember that meiosis has the 1 round of DNA replication before it begins, just like mitosis. So you have the duplication of DNA. But then, since there are 2 rounds of cell division in meiosis, that means that we're going to end up with half the amount of DNA that we would have if we were just going through mitosis. So again, if you think about this, if you think it through, you say, okay, mitosis, you start out with double the DNA, and then you end up with half of the double, which is just a normal amount of DNA or a single genome. And in meiosis, you start out with double the DNA, but then you go through 2 rounds of division. So you're going to go down to a single genome, and then you're going to go down to half of a single genome. And that's what the resulting cells end up with. So that's shown here. In this schematic, we start out with 1 cell with 4 chromosomes, and then during S phase, there is DNA replication. And now in this cell here that has not yet begun meiosis, we see a duplicated genome, so twice as much DNA as we normally would see in this organism. And here we have homologous pairs of chromosomes. So let's take a moment to talk about some vocabulary. Here I'm showing 2 different sets of chromosomes, so just ignore the text for now, but I wanted to show that the blue chromosome is from one parent. We'll mix it up. We'll say that the blue one's from the female, and then the red one is from the male parent. So you could compare this to you having DNA from both of your parents. And each of these singular lines that's making up the X is an identical copy. So we see that from the mother here, we have some gene variant that is a capital A, and then another gene, and it has a capital B, and then we see the exact same thing on this other line here. And so these 2 are chromatids, they're sister chromatids, and this whole thing is considered to be a chromosome. And then we have this chromosome from the other parent. As we go along with our analogy, this might be from your father, and this has the same genes, but not the same variants on it. So we see here that we have the lowercase a variant and the lowercase b variant. So we have different variants for both of these genes. But we do see, again, on this chromatid on the left and the chromatid on the right, they are identical, okay? And then, so all together, the blue and the red chromosome, they are called homologous chromosomes. So homologous means like or comparable, and so you could think of these as being comparable but not identical, because again, they're from different parents, so they have different variants, different DNA sequences, but they code for the same genes overall. So if your mother gave you brown eyes and your father gave you the gene for blue eyes, you're getting a gene for eye color from both parents, but you're getting different variants of that gene. All right. So to come back to here where we left off, again, we have homologous pairs of chromosomes here. And so there are two different chromosomes overall in this organism. We have one that's a larger chromosome and then a smaller chromosome. And then we have homologous pairs of both of those chromosomes. Then we go through meiosis, the first division, and we see that the daughter cells from the first round of meiosis have the amount of genetic information, but we do not have it in such a way that we have a complete copy of what we had in the starting cell. So we notice that the daughter cell on the left is now missing the homologous chromosome for the larger chromosome of the blue, or the male, going along with our analogy. And likewise, this cell on the right is missing that larger chromosome from the female, the red one. And then we go through the second division, and this is where we separate out chromatids. So the second division is almost more similar to mitosis, which we talked about yesterday, in that we're separating the chromatids. And the first round of division is different from what we talked about yesterday, because in that round, we're separating homologous pairs rather than chromatids. So really make sure that you have an excellent grasp on the difference between a homologous pair and sister chromatids, because that's going to be really crucial for moving forward in this lecture and for the quizzes and exams and your career in biology in total. Because that can get very confusing. All right, and then I want to point out one other thing, or two other things, which is that the terminology for the type of genome that is seen here is diploid. So diploid means that we have homologous chromosomes, but we have only one chromatid for each chromosome. Then here at the bottom, after both rounds of meiotic division, we have haploid genomes, and that means that we only have one chromatid from each homologous chromosome. So if you think about it in terms of eye color again, then we might have a variant for brown eye color and a variant for blue eye color, and both of those are present in the same cell. And then we go down to here, and in this cell, we only have the variant for brown eye color. In this cell, we only have the variant for brown eye color. And then in these two cells, we only have variants for blue eye color. So again, haploid, we don't see that the different variation because these cells have only one copy of the pair that makes up the homologous chromosomes. All right. And also here, so a diploid cell is called 2N, and that means we have two copies of every locus. So another term, locus, is just a position along the DNA. And then when we go here, we have a 4N cell, because we've doubled the amount of genetic information, the amount of DNA. the mass of DNA that we have, going from our 2N, going through S phase, and going into 4N. And then we eventually are going to have just N, or haploid, in the resulting daughter cells. And so I do want to mention, in reality, so this is the just overall schema of the process of meiosis, but in reality, we don't always get 4 daughter cells. So in sperm production, in males, you would end up with 4 daughter cells from this one original cell. But in egg production, there is an unequal distribution of cytoplasm. So we end up getting only 1 egg cell, and the other cells end up being what are called polar bodies, and they're very small and they can't be fertilized. And so I just grabbed a picture to show you guys here. So again, in the process of meiosis for egg cells, we have the 2 divisions, but these 3 cells end up being much smaller. So they do have the DNA in them. They have the appropriate aliquoted DNA, which is only going to be haploid, but only one of the egg cells ends up, or the daughter cells ends up being viable as an egg. Whereas we have a comparison here in the meiosis that results in sperm cells, all 4 of the daughter cells ends up being a viable sperm cell. Okay. And I also wanted to mention really quickly that I've labeled this here as 2N or diploid. I think your text disagrees with that. I'm not going to ask you either way because I've seen it both ways in the literature. So don't get too hung up on that. Just know that even if you're calling this 2N, it's different than the 2N that we saw originally, because we don't have, obviously, these are not identical to one another. Okay, so this is just an image that lets you look through every single stage of meiosis. And so we talked about mitosis here on the left, and we talked about all the phases in mitosis. So prophase, metaphase, anaphase, telophase, and then we see the actual division of the cell material and in cytokinesis. And so we go through all of those phases in the 2 divisions of meiosis. And so that's what is shown here on the right side. We see prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. And then we go through all of them for a second time. And that's how we end up again with these 4 daughter cells instead of the 2 daughter cells that we see in mitosis. And so I think that this is also a really useful slide for reference. So I recommend definitely going back to this and using it to study a bit, because it lets you see the normal human karyotype during G1 phase. So in other words, during the regular growth phase when the cell is not preparing for mitosis, and then a normal human karyotype after S phase or during G2 phase. So actually, it's not really in S phase, because it's in S phase that it is copied. So this is in G2 phase before mitosis or meiosis. And so again, you could compare here using the correct terminology that we have 1 chromatid per chromosome when we're looking at G1 phase, and each chromosome has 1 homolog. In other words, you get 1 chromosome from each of your parents. And we see here, this is the fictional or cartoon karyotype or layout of chromosomes for a human. So we have our 23 different chromosomes. There are different genes on all the different chromosomes. And again, if we look at chromosome, actually I believe it's chromosome 2 that carries genes for eye color, and here we might have brown eye color variant, and here we might have blue eye color variant. And then none of these other chromosomes are going to carry variants for eye color because that gene only occurs once in the genome. All right, then we look over here after the genome is duplicated after replication, and we see that for chromosome 2, now we have 2 sister chromatids per chromosome. So this purple X is 1 entire chromosome. And then this blue X is 1 entire chromosome as well. And we see again that we have 2 homologs as to make up a pair. And so I've stated that here. So we have twice the amount of DNA. And then in this instance, if we were looking at we were trying to count the number of brown eye variants, or the gene for brown eye variants, we'd actually see 2 in this cell because this chromosome 2, this homolog, has 2 exact copies. Whereas we would only see that brown eye variant once in G1 phase because we only have 1 copy of it here. One last thing I'd like to point out is that all of these chromosomes 1 through 22 are called autosomes, and these are non-sex determining chromosomes. And so that's most of our chromosomes. And then the sex chromosomes in mammals are the X and the Y chromosome. That's the 23rd chromosome that we see here. And we use the same terminology for the duplicated genome. So another difference between mitosis and meiosis is that during meiosis 1, we have a process called crossing over. And this occurs when the chromosomes, the homologs of the chromosomes line up at the center of the cell. So in mitosis, we have an alignment that looks like this. This is also a really great slide to study from, and you should be able to draw this out, the difference between the lining up of chromosomes in mitosis and meiosis 1. So in mitosis, we draw this out so that we're going to end up having 1 chromatid go to each side of the cell. So this one on the left is going to go this way, this one on the right is going to go this way. In order for each daughter cell to end up in this version after the first round of meiosis, then we need to line up the chromosomes in this way. We line up the homologs next to one another. So we now have the homolog from the maternal parent and the homolog from the paternal parent. And then down here, we have the other chromosome. So going back to this slide, we are seeing that we get both of the homologs for chromosome 1, and they are next to each other, and then they're going to be pulled apart. And as they are pulled apart, that involves 4 chromatids, and you're separating 2 chromatids and 2 chromatids. Whereas in mitosis, you have 2 chromatids to start out, and you're pulling them apart into 1 and 1. So again, here we have 2, we're pulling apart into 1 and 1. Here we have 4, and we're pulling apart into 2 and 2. And you can see why that is going to reduce the amount of genetic information in each resulting daughter cell. And we also see why we end up missing gene variants. So for example, if only the mother had that variant for blue eyes, and this entire homolog is going to go to this daughter cell on the left, the father has the variant for blue eyes, then it's going to go over to the right, and that cell is not going to have the variant for brown eyes. So this structure, or this layout, allows for something called crossing over, which I just mentioned. And during crossing over, there is the actual physical exchange of DNA between chromosomes. So again, we're just dealing with chromosome 1 lining up with other versions of chromosome 1. So we're not swapping genetic data between, chromosome 1 and chromosome 3, or chromosome 3 and chromosome 20. You could see why that would not work because, for example, chromosome 3 is much larger than chromosome 20, so you would be ending up with an imbalance of genetic data in the daughter cells. Instead, we line up the appropriate analogous or homologous chromosomes, and genetic data is exchanged, or genetic material is exchanged between these homologs. In theory, it's also possible for the chromatids to exchange genetic material, which would not really generate much diversity. So the whole purpose of crossing over is to generate a single chromosome with more diversity than the original chromosome had. So the way that this happens is that the chromosomes, the homologous chromosomes, are held together by something called a synaptonemal complex. And so it's just a complex of proteins that, again, forms kind of a mesh structure, and it lines up the DNA down the length of the chromosome. And then we actually have the cutting of the DNA backbone and on two pieces of DNA, and then the exchange and reattachment of the DNA. And so we can see here, again, a cartoon of two chromosomes. And we have homologous chromosomes, which are lined up linearly. And here we have a chiasma, which is just the point where the genetic exchange occurs. And we are going to break this blue chromosome here. We're going to break this red chromosome here, then exchange the genetic material. And so the result is going to be something like this. So we see that we have this chromatid on the left, and it has mostly genetic material from one parent, but then due to crossing over, it's going to have some genetic material from the other parent. And I apologize for jumping around a little bit, but you could see on this slide here that you could have multiple chiasma in a single pairing of homologous chromosomes so that you could get lots of crossing over. Although the closer together two loci or two locuses are, the less likely you are to get a crossing over event occurring between them. So if we were looking at a loci that was right here, a locus that was right here, and then a locus that was right here, it's not very likely that a chiasma is going to form between those two because they're very close. If you're looking, on the other hand, at a locus that's way up at the top and a locus that's way up down at the bottom of the chromosome, it's very likely that there are going to be chiasma forming between those two and crossing over events. So one more term here is bivalent, and bivalence refers to the entire complex of the two homologs lined up with one another. So that's two homologs, 4 chromatids overall, that is one bivalent. So in this image, all the way on the right here, we see 2 bivalents. Okay. And then here, not going to go on Kahoot right now, if we had these, chromosomes at the top, these homologous chromosomes, and one parent had the lowercase variant of the gene for A, and the other parent had an uppercase variant for the gene for A, I'm asking, when would crossing over not result in a novel genetic sequence? So take a moment and think about that. If we have these 2 homologous chromosomes, they form a bivalent. How could you have a crossing over event, but no novel gene sequences, no novel combinations of genes or variants on the resulting chromosomes? And so that would occur if we had the swapping of genetic information between chromatids, as I mentioned before. So that can occur because, this is all happening kind of mechanically through molecular molecular mechanisms, and there's nothing to stop the two chromatids from exchanging genetic information rather than the homologous chromosomes. So that is a possibility, and it kind of defeats the purpose of crossing over in meiosis. So let's go back to this cardinal that's half male and half female. So this is most likely a case of a mutation, but not the normal kind of mutation where you change a single base pair or a few base pairs. It seems like this is the improper segregating of either entire chromosomes, or as it's suggested here in the text, it's suggested that the egg cell actually had two nuclei and carrying one chromosome for each of the different sexes. And so in birds, there's no X and Y. Instead, there are W and Z chromosomes. And the nuclei, we think, had, one of them had a W chromosome, the other one had a Z chromosome. And then when fertilization occurred, then we ended up having one nucleus that was male and the other nucleus which was female. And then after the fertilization occurred, then of course mitosis from each of these two nuclei resulted in a line of cells, or a lineage of cells, that shared the sex of that starting nucleus. So I wanted to just lay out the entire sequence for when an organism is producing germline cells, what they produce, and then how the germline cells are going to get back up to a diploid cell. And so we go through meiosis, and then that's the resulting cells are gametes, so either sperm or eggs, or in the case of females, sometimes polar bodies, which end up not being used. And then there's fertilization. And through fertilization, at that point, that's where we get back up to 2N. So meiosis results in N, and gametes are N. So in other words, they are haploid. And fertilization then results in 2N, or diploid. And then after fertilization, we have mitosis, and that's 2N, and it stays at 2N. And so that's the sequence. Okay, so I think that we're done with meiosis. I encourage you to go back and review, and I'm going to move on to DNA replication, but I think that I'm going to start a new video for that. So goodbye for now.